Alrighty, hello everybody. Today we're looking at Lecture 8, which is talking about chromosome structure. And yeah, let's go ahead and get into it. Your exam is actually this Wednesday, so recording this on Sunday, so guys have got a couple of days to watch this if you want and get your practice in if this is what you've been waiting for to do practice for whatever reason. But yeah, just a real quick recap of Lecture 7. We talked about DNA polymerases, we talked about the catalytic mechanism, their structure, how it looks like a hand. We talked about chain terminators and their use in things like Sanger sequencing. And then we also talked about how there's like proofreading in the polymerase, like the 2', like how DNA polymerase can't recognize a 2' hydroxyl for the normal NTPs, so it has to have the DNTPs. So stuff like that. We also talked about Sanger sequencing and next-gen sequencing. I believe these 2 points should be switched. Next-gen sequencing is used to measure multiple things at once, while Sanger sequencing is usually just looking at one molecule of DNA. So, but don't quote me on that. But yeah, I mean, everything else is fine. Use single analyte. You can use many different DNAs per thing. You can use irreversible chain terminators for Sanger and reversible for next gen. This is the most important box, I would say, for sure, is like what kind of terminators are you using, and that's the important thing to remember. So you use irreversible for Sanger and reversible for next gen. If you were to take one thing away from Lecture 7, it would have been this. Okay, now we're going to move on to chromosome structure. I'm not going to like bog you down in all the nonsense, and there's a lot of words in this, but yeah, we'll get through this quickly, hopefully. So the difference between a genome and a chromosome and chromatin. So genome versus chromosome versus chromatin. So genome is like all the genetic information. So it's like everything in the cell. So like all of your genomic DNA and all that stuff codes for like, I don't know, proteins and tRNAs and rRNAs and all that stuff. That's your whole genome. So a genome is just everything. A chromosome is a large DNA molecule that houses part of the genome. So you have multiple chromosomes. So you, for example, humans have 46 different chromosomes, and all 46 of those chromosomes make up the genome of the human, if you will. So a chromosome is a single unit of the genome, if you will. And then the chromatin is kind of like a complex of DNA, histones, and other proteins that kind of it makes up a chromosome. So you've got histones surrounded by DNA, and that DNA coils up, and that's what chromatin is. And then when chromatin combines and kind of clumps up, you get a chromosome. All that to say that each of the chromatin makes up a chromosome, and then chromosomes make up genomes. So it basically goes like this. And chromatin is just the DNA plus any binding proteins that hold it together. And then a chromosome is just multiple units of chromatin, and then a genome is just multiple chromosome units stuck together. Or not stuck together, but combined. So this is just a cool image of what bacterial chromosomes look like. Cool, don't think it's that important. What do they look like in eukaryotic cells? If you've never seen mitosis before, this is what mitosis looks like. Specifically, this, I believe, is the anaphase. This is anaphase of mitosis, if I'm not mistaken. And this is a telophase. Telophase are getting close to cytokinesis here. So the cool thing about chromosomes is, in eukaryotic cells, is that it seems like they actually kind of move to different areas of the cell. So you can see here, they show dynamic structures in different cell states. So like at different points in the cell cycle, they're actually in different places in the nucleus and stuff like that. So you can see like all of these numbers, right, represent the chromosomes. So chromosome 1, you got one copy of chromosome 1 here, and then the other copy of chromosome 1 is over here. So chromosome 1, you can see your X and Y chromosome here. So, they kind of organize themselves in a weird way within the nucleus. So, something cool to keep in mind, I guess, but I wouldn't be too worried about that for your exam. So, the other thing is that, chromosomes and DNA are usually, within the nucleus, they are bound using these sorts of DNA binding proteins. And you can actually see those like here. So these binding proteins kind of help bind the DNA to the nuclear lamina so it doesn't just float around mindlessly in the nucleus. So, and also just to emphasize this, chromosomes are always decondensed unless they're going through mitosis, meiosis. So this whole image here, this type of chromosome, you only ever see this during mitosis and meiosis. You will not ever see that kind of chromosome when you're just in a cell that's at rest, right, in like G1 phase. It's only when it's undergoing mitosis and meiosis does it condense like that into that X shape. But here we're going to go through different parts of DNA that make up a chromosome, if you will. So if we look at this, we have our double-stranded DNA here. And then, that forms a double helix. And what you can do is you can take histones. You can take 8 of those histones, form this, cube with them. And you can wrap your DNA around it to form a nucleosome. And then you can take these nucleosomes, you can add them together to make chromatin. And then this is chromatin here. And you can basically wind up the chromatin over and over again into a chromatid. And then the chromatid, you basically do this a billion times, and you basically stack it on top of each other, like bang, 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 whatever, right? And you make a chromosome. So the chromosome is I would say, I guess is the largest form of, I don't know, DNA, like a DNA structure that you'll see, where, all of the DNA is consolidated into this one large X-looking structure. I'm not going to bother with this video, but you can always watch it on the normal lecture on your own time. But let's go ahead and take a look at how nucleosomes are assembled and how the modifications work. So the first thing to look at is chromatin. I already talked about this. Chromatin basically looks like beads on a string. So remember, each of these little balls here is going to be made up of 8 different histones surrounded by a bunch of DNA. And this is a number that could be useful to remember, 11 nanometers. So there's 11 nanometers, I believe, between, I guess you were to take the distance between a nucleosome and another nucleosome. It's 11 nanometers, I believe. Or more specifically, on either side, there is, if you were to take the space that a single nucleosome occupies, it's going to look like around 11 nanometers or so, seems like. Nucleosomes are not rigid, so yeah, they can slide along DNA. So it's not like it's tightly wrapped, right? I mean, they can move and they can shift around. So you can imagine maybe it uncoils a little bit and slides a tad bit, but it's nothing too crazy. And then the other thing is that histones, right, these proteins actually introduce negative supercoils. And so what happens is you get this histone. This histone slides into this pocket here, and then it kind of wraps the DNA around it. And then this kind of weird wrapping is fine when you have the negative supercoil, but it's actually kind of unfavorable if you have this positive supercoiling with this negative supercoiling. So what actually ends up happening is you actually end up using topoisomerase to get rid of that negative or that positive supercoiling, so it actually becomes more favorable. So yeah, and another thing to note is that histones are the most common protein in nucleosomes, but type 2 topoisomerases are actually also similar in abundance compared to histones. which is kind of insane to think about, right? That just means that there's actually a lot of this kind of stuff going on here, where because you're binding, or you're trying to bind so much of your DNA to your histones, you actually end up forming a lot of these positive supercoils that make it unfavorable, and so therefore you actually need a lot of topoisomerases to make sure it remains stable. And the binding of the DNA to the histone is actually very stable because the histone is actually positively charged and the DNA is negatively charged, so there's actually a charge-charge interaction there that allows it to kind of bind happily. So there's like negative charges all over this histone. Oh, sorry, I think I messed that up. There's plus charges on the histone. There's plus charges all over the histones, and then there's minus charges all along the DNA, right? That's not how minus charges look, but you get the idea. Minus, 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 minus. There's a bunch of minus charges, right? And so these can associate and form these kind of complexes, and that makes it very stable. So that's why it's actually favorable in the first place to even bind this histone in the first place, despite the fact that you're forming this unfavorable positive supercoils, which you can just use topoisomerase to remove. Okay, so for the see, just open that. Don't need that. Okay, for the nucleosome assembly, essentially, I would say this is something you should probably put on your note card, but basically what you're going to do is you're going to have a couple different types of histones. You got histone 4, histone 3, histone 2b, and histone 2a. And the histones are composed of a lot of lysines and arginines. More than 20% of these histones is made up of... So each histone basically is made up of amino acids because they're all proteins, but 20% of the histones, each of the histones, or at least 20%, is made up of lysines and arginines, which are both positively charged, right, at neutral pH. So then what you do is you basically take these things and you form dimers. So H2B and H2A you combine to make a couple heterodimers, and then H4 and H3 also combine to make a couple heterodimers. And what you do is you basically take these and you stack them on top of each other, and you take the H3, H4 dimers and stack them on top of each other, and you combine them all, and you make this. And this is an 8 subunit complex, which is a nucleosome once you wrap the DNA around it. So it's an octamer, right, of histone proteins, and you wrap the DNA around it, and it's a nucleosome. And here's that 11 nanometers again. This kind of structure here is 11 nanometers. I think above I said that the distance is 11 nanometers. To be more clear here, this thing is 11 nanometers. The length of this is 11 nanometers. And so the DNA wraps around the octamer around 1.67 times. And the number I would keep in mind and try to remember, or the couple numbers I try to remember, are maybe this number, how many times the DNA wraps around, and for sure this number. You should absolutely remember or memorize that there are 147 base pairs of DNA that are able to wrap around a nucleosome. That is something you have to remember. Because when you do gel electrophoresis and you use things like DNase or like these different restriction enzymes to cut DNA up, if there are nucleosomes, you'll actually get bands in like multiples of like 150 or like 200, because of the fact that 147 of those base pairs are stuck on a nucleosome. So you can't actually cut it because the restriction enzyme or the DNase can't even reach it, since the nucleosome is blocking enzymes from getting to them. And I say it could be like for 150 to 200, or maybe even more than that, is because there's also like little edges that hang off. So imagine this is your nucleosome, right? And like there's a protein in the middle here. There's also the parts that overhang off, right? So let's say that you cut with restriction enzyme here and here. There's still like this whole section here of DNA that you have to keep into account. So like this over here is 147 base pairs, but maybe over here there's like 25 base pairs and another 25 base pairs on this side. So you can see how it could get to like 200 really quickly. What happens if you leave this in a gel for a long time, though, is like, you know, eventually if you do have cuts here initially, eventually it'll cut smaller and smaller, right? And it'll eventually get into like the point where it's exactly 147 base pairs if you were to run it for time equals infinity. But I think before then your histone would probably break and break down before you even ever got the chance to see that it's exactly 147 base pairs. So. So here's a little image. I don't think this is that important. I think maybe this is important to know. So there's about 140 hydrogen bonds between peptides and the backbone of DNA. So that's something to keep in mind, right? So that's how you stabilize the DNA to the actual histone. So in that plus the charge interactions as well, right? That's important. So there's hydrogen bonds between the peptide backbone and the DNA. And there's also the, this is the core motif of what a histone has. So it's got one alpha helix, a second alpha helix, and a third alpha helix in this kind of like U-shaped pattern. And, you know, do I think that's that important? Not really. Maybe something to put on your note card, but I think this is more important than what is the exact core fold motif. So. Yeah. The other thing you should be able to tell is actually look at all of the plus charges, if you will. There's like arginine. I can't really see anymore. There's going to be a lot of like arginines and lysines if you were to go through this whole thing. Lysine, arginine, arginine. So yeah, you can see if you go through all this, there's a bunch of like things like arginine and lysines that are there. And those are going to be helpful for, you know, the charge interactions. And there's other amino acids that are helpful for the hydrogen bonding. Yeah, so I think the adjacent minor groups are... I don't think this is that important to keep in mind. Yeah, I would say that this is not the most important thing. This here is not the most important thing. Just know that they're flexible. Even I'm not entirely sure what this is supposed to be getting at. But do channels formed by adjacent minor grooves, adjacent helices. By adjacent minor grooves of adjacent. Yeah, I don't know. I feel like that's not. I don't think it's that important though for this. I think there's other things you could be tested on. But yeah, I think it's important to know that just like these histone tails, they're kind of floppy. They don't like they're not very stagnant and stuck. So they're able to move around and flap around a lot, which actually makes them very vulnerable or very easy to manipulate through enzymes, which we'll talk about in a second. And also you talk about a lot in 442. So the other thing that's important with the tails is that they actually allow for contact between histones. So you can see here like these histone tails are actually connecting with each other and they're interacting. So they actually allow for the histones to organize with each other, or the nucleosomes to organize with each other. And remember that each histone has its own tails, right? So the 2A, 2B, 4, 3, these all have their own tails. And so the tails allow different nucleosomes to interact with each other. And kind of form these higher order chromatin structures where like they get compact and get stuck together. And so the other thing to keep in mind about them is the way that we're able to modify them. And there's many ways you can modify them. You can modify them through things like methylation, acetylation, phosphorylation, and ubiquitination. Ubiquitination, I'm pretty sure, is for protein degradation, so that one's a bit later on in the course, if I'm not mistaken. But things like methylation, acetylation, and phosphorylation, those are things that you guys should be like relatively familiar with. Like maybe acetylation and methylation is a bit weird, but you guys know what methylation is from OChem, and acetylation is just when you add this kind of group to something. So you can imagine that you basically take some sort of group and you add that on there. So this usually, so the ones that you actually care about, I would say, are going to be these two. And what's important about those is, remember how I said, you know, that DNA is negatively charged and the histones are positively charged? Well, if you acetylate these lysines, right, so these lysines are positively charged, if you acetylate them, it actually makes them neutral. And so that interaction of positive and negative is no longer there, and that results in the DNA actually unwinding from the histone, if that makes sense. So essentially there was a charge interaction before of negative and positive, where the histone was positive, but by acetylating it with an enzyme, that interaction is lost, and so the DNA is able to unwind pretty easily. And so that's something that's stated right here, actually. And so there's some enzymes that we use. We call them writers, readers, and erasers. So writers are enzymes that, if you will, they write on the DNA, or they write on the histones, not the DNA. They write on the histones, and they write on the histones by adding an acetyl group to the histones to make them more neutral. So things like histone acetyltransferase is a writer, or histone methyltransferase is another. I think methyltransferase enzymes are another example of a writer. Reader domains are enzymes that read, or like are able to bind to those modifications. So say, for example, that you have an acetyl group that's attached to a histone, so some sort of acetyl group is attached. There might be an enzyme that binds to that addition of an acetyl group. So maybe that acetyl group is something that's required for an enzyme to bind, like a bromo domain. This is part of an enzyme, or part of another protein. So those are readers. They're readers because they read the modification, they bind to it. And the eraser does exactly what it sounds like it does. Histone deacetylase does the opposite of acetyltransferase. It removes the acetyl group and restores the plus charge. So it literally erases the thing that the writer did. So hopefully that makes sense. It should be relatively intuitive, I think. But yeah. Okay, so the difference between heterochromatin and euchromatin. Heterochromatin is closed off, and you are not able to do any sort of binding, transcription. You can't do anything like that. But euchromatin is the open form, and euchromatin is the way that we're able to do like transcription and stuff, and able to do things with the DNA, including transcription and going into translation and all that stuff. So if you want to transcribe a gene, you cannot do it while it's in the heterochromatin form. It's got to be in the euchromatin form, where it's open. So yeah, the amount of euchromatin and heterochromatin distribution changes depending on what stage the cell is in. This should make sense. When the cell needs to, you know, if certain types of cells are going to need certain genes to be more active than others, and therefore they're going to have those areas be more euchromatin-like than heterochromatin-like, right? So for example, something like a granulocyte. So granulocytes, you can think of them as let's see here. You can think of them as like immune cells, right? So they're going to want to produce proteins that are useful for immune response, right? And so they're going to have regions of the DNA that represent or that have proteins related to the immune response in euchromatin form and not heterochromatin form. But they would have, genes that are related to like digestion, right? In heterochromatin form, because they're not responsible for digestion. They're responsible for defense mechanisms. Similarly, for hematopoietic stem cells, so like for things like your liver, right? Your liver is going to want enzymes or proteins associated with digestion and like glucose regulation. They're not going to want sections of the DNA that are important in immune response, right? So these stem cells, these liver cells, basically, are going to have the sections important for glucose regulation in euchromatin form, and the sections for immune response in heterochromatin form. So what's in heterochromatin form, what's in euchromatin form depends on what the cell type is, and what does it need active. This is just a nice little image that says that there are other ways to assemble nucleosomes. So we talked about how nucleosomes are usually these 11 nanometer blobs, but you can also assemble them in like these little triangles. You can assemble them in squares. You can assemble them in like all sorts of weird little shapes. So you can connect them together. Like you can see here, there's like 3 of them. It's just a cool way of saying that, you know, there's a bunch of different ways that these things can assemble. So here's some example of that. There's tetranucleosomes. There's 4 of them. So 1, 2. There's 1 here. 1. There's 2 here. There's 3, and then there's 4. Right? 1, 2, 3, and then there's a 4th one underneath. And then you have like this. This is, for example, is actually how chromosomes are formed. Chromosomes take this form of the nucleosome / chromatin, and then they kind of bunch it up together, and then they combine a bunch of them together. There's also one last important part of the histone that is there, and that is going to be the H1 portion, the H1 protein, which is histone 1, I believe, if I'm not mistaken. And histone, this H1 is actually very important for stopping the thing within the nucleosome, the things within the nucleosome from being transcribed and accessed. So, unlike the other, you know, H2A, H2B, H3, H4, H1, there's only one of them per octamer. So, and that makes sense because, you know, it kind of sits in the front here, and what it does is it basically acts like a belt, if you will. It basically binds to the DNA and, like, tightens it onto the nucleosome. And so it makes sense that, you know, if you're tightening it, it's going to be harder to access. So you're not going to want that many of them, because if you add too many of them, it's going to be impossible to access the DNA. So there's 1 per octamer. There's a bunch of variability with them. There's, you know, 8 different subtypes of them. But the main thing to note from these is not only is there 1 per octamer, but they also decrease the ability of other enzymes to bind to the DNA. So that's the important thing to note. And then you can see here that, you know, when H1 is bound, this is the other thing I'd actually add to your note card. When H1 is bound, 168 base pairs are protected, while when it's not bound, it's 147. So, you know, I remember I said to put this note card down on your note card or memorize it. This is the other one I'd memorize, because, you know, Goo loves, like, the fill-in-the-blank question, so he could totally ask, when the nucleosome is protected by H1, this many base pairs are protected, or when it's not protected by H1, this many base pairs are protected. So, just something to be aware of, and that, you know, it's fair game on the exam. And the other thing to remember is that, you know, regions that are like being actively transcribed, they don't have H1. And that makes sense because, you know, H1 tightens it up, makes it harder to transcribe. So if you have an area that is actively being transcribed or needs to be transcribed, you don't want to put H1 there, because H1 is going to make it harder to transcribe. This is just a cool little section talking about, you know, other proteins. There's other proteins there available that can do this kind of compaction method. HP1, example of that. The H3K9 method. This notation you'll learn about in 442, but it's talking about basically histone 3, lysine 9 has a methyl on it. You'll learn what that means, but there's heterochromatin protein 1, which is HP1, and heterochromatin protein 1. Believe it or not, heterochromatin means the closed-off form of chromatin, so these things help compact the chromatin more, if you will. So there's a whole bunch of these things. I don't think it's that important to know for this class, but, you know, this beads on a string structure is just a very fancy way of saying nucleosome, section of DNA, nucleosome, more section of DNA, right? So we've got section of DNA, nucleosome, section of DNA, nucleosome, section of DNA, you know, yada, 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 yada. It just keeps going, right? Yeah, so this is actually a really cool experiment that you could do. Long story short, basically what they're doing is they're taking a chromosome, right, that's in its condensed form, And then they're looking for the regions of the chromosome that are, you know, kind of open and available to be chopped off from the nucleosomes. Like you can see this small section here, there's a section here, stuff like that. So they use these like hyperactive TN5 transposase enzymes. I don't think these are, it's important to know what these do exactly, but basically what these things do is that they open up the chromatin. And then what they do is that they fragment the DNA, right? And so when they fragment the DNA, you'll get the sections of the DNA that are like actually able to be bound to by enzymes, while everything around the different nucleosomes is like stuck. You can't bind any of these, right? Because all the DNA is stuck together and tightly bound to the histone. And so you can't do anything with that. And then what you do is you wash away those histones, the nucleosomes, and then you just continue to PCR the sections of DNA that you do have with you. And then what you can do is you can then next-gen sequence those DNA fragments that you get, and you can map them to a genome of the organism that you're looking at and the chromosome that you just sequenced, and you can see which regions of the chromosome are actually open and able to be transcribed. And that gives you an idea of, you know, maybe what kind of cell is this, right? So certain cell types are going to have certain genes that are active and in the euchromatin form. And so you'll notice that different cells actually have different profiles, right? So this cell, whatever this cell is, has this profile. But, you know, maybe I have a cell that has a profile that looks like this, right? Maybe there's something up here. You know, maybe this section of chromatin's actually open, right? Maybe there's nothing here, and nothing here, and maybe there's like a section here. So you can imagine, like, if you draw something like this, right? You know, maybe here. So maybe you have a graph like this, and there's like a lot of here. Maybe there's a lot of transcription activity here, and there's nothing else for the rest of it. So this here is a different cell. This image I drew here is a different cell from this image, because you can tell by the sequencing profiles. You know, over here there's nothing over here in this cell, so these are different cell types, and they require different genes. So, yeah. I hope that makes sense. Bit of a shorter video, I think, today. It's actually probably around the same length. But, yeah, I hope this lecture was helpful, or this review was helpful. If you have any questions, feel free to stop by the IC. We'll be doing a exam review workshop on Tuesday, I believe. But, yeah, other than that, good luck on your exam, guys, and happy studying.